So welcome to our SPRINT Toolbox webinar. My name is Vera Silva. I'm from Wageningen University and I'm the project manager of SPRINT and will be your host today, or moderator of the event. We have quite a big agenda to share and to discuss with you. So we'll be starting in like a minute or so. Just giving a bit more time so more colleagues can join us in the event. In the meantime, I encourage everyone to use the chat to leave short information about where you are seeing us and why you are seeing us. As you may notice, this is a webinar type of setting, so your camera and your microphone are not open, but we do encourage you to use the chat for communication and also the Q&A display to leave questions for our speakers, because we will come to that in several moments of this event. Again, I see the numbers are rising. Welcome everyone to this SPRINT Toolbox launch. Yes, okay, so 45 already with us. Yeah, also be mindful that this event is being recorded. Again, the cameras are off and this will come shortly after the event. We will display this recording in our SPRINT YouTube channel, so you can revisit the content and also answers and questions posed during this event. Yeah. And all those that we don't have time to manage here during this session, we will do afterwards via email. So don't feel shy, just push your questions to us in the chat and the Q&A box. I see that someone cannot hear me. I hope this is a punctual case. Can I see any nodding heads for me? The audio is fine? Yes? Oh, okay. Yes. Okay, well, again, for those that just joined, welcome. Thank you very much for joining us here. Extend a warm welcome from all the SPRINT Consortium and also the colleagues on the back end to support this event. My name is Vera Silva. I am here manager of the project and will be your host today. I will show you the agenda of the event and guide you a bit through the different tools that we have prepared during the last 5.5 years. So stay with us and use the chat and the Q&A box to leave your questions. We will also use the chat to leave you some links and some references to support and to provide additional information on the tools that are being presented. Okay, so please keep a close look onto this. Okay, then I would say this. We are now 62 colleagues. I see people are still a bit shy in the chat to leave their names and their locations. Please do use it. We would love to hear from you. Yes, and also indeed expectations. So we hope that we can have the time to address them all. Yeah, I see that is basically time to start already with content. So that is why you are here. So I think I will already start giving the floor to Jakob Hoffman, who together with Anke coordinated all this toolbox work that you will hear all about during the 90 minutes to come. Yeah, Jakob, please.
Okay, dear ladies and gentlemen, it's my great pleasure to welcome you to the official launch of the SPRINT Toolbox. This platform is one of the project's flagship outputs, the result of several years of intensive collaboration by researchers from organizations across Europe. I would like to, if you can go back, Jane, I would like to also take this opportunity to express my deepest gratitude to everyone who did hard work and made this possible. So you can see the names here. Next slide and next slide. And yes, this one. Thanks. The Global Health Risk Assessment Toolbox, it was developed within the European Horizon 2020 project SPRINT. What it is an open access web-based platform with 10 interlinked tools for integrated exposure, impact, and risk assessment of pesticides and their mixtures. The toolbox combines outputs from different SPRINT work packages and spans from the local case study sites to the pan-European level. Next slide, please. So what does the SPRINT Toolbox actually contain, what it can do? It brings together three core layers of information. First, it offers interactive presentations of the SPRINT results. So that means case study findings, toxicity and ecotoxicity studies, human health impact assessments, and so on. Second, it also provides modeling, upscaling, and mapping approaches for pesticide occurrence, exposure, and risks. And third, it includes also data mining, analysis, and extrapolation tools, drawing on national and European pesticide dataset. So you will learn during this seminar about individual tools. The toolbox, in short, streamlines the SPRINT project's impact into a single assessment hub for policymakers, researchers, and the public. The primary end users of the toolbox will be decision makers and policymakers at national and EU levels, regulatory bodies. It means, for example, European Food Safety Authority, EFSA, European Commission, national authorities, then risk managers, agriculture stakeholders, agri-food industry, and environmental scientists. But it may also be used by the general public, which is interested in pesticide exposure and impact. The toolbox supports all of them in understanding pesticide exposure and risk, and to improve their access to data, models, maps, and results on pesticides occurrence, fate, and effects. So that was for the introduction, and now we can go tool by tool in 3 blocks.
Thank you very much, Jakob, for this introduction and overview. Indeed, I would like to, without further ado, to give the word already to Rosita and to Paula to show results that we got in the field and then in the lab. Rosita, please.
Thank you, Vera. Good afternoon, everyone. Today I will briefly introduce the case study sites result tools, focusing on how users can explore and navigate the data. Next slide, please. The case study site results tool presents the main results of the SPRINT monitoring campaign. It shows where pesticides were found across the case study sites and what the potential ecological risk is in the soil and water. The tool is based on the data collected during the large SPRINT monitoring campaign in 2021. In total, more than 200 pesticides were analyzed across environmental, human, and animal matrices. In the introduction page, as you can see in this slide, users can find background information on the list of analyzed substances, the limits of detection, the cropping system, and livestock present at each case study site. So overall, this tool provides both the scientific background and the visual exploration of the results in one place. Next slide, please. In this slide, you can see how the data from the 11 case study sites are structured. The monitoring campaign covered 3 main matrices: environmental, human, and animal. In the figure, you can see which compartments were analyzed within each matrix, for example, soil, water, dust, and others. Users can navigate directly to any of these sections through the left side menu in the toolbox. You may also notice some gray blocks in the overview tables. These gray blocks indicate the samples that they were collected during the SPRINT campaign, but are not represented in the tool, mostly samples used for the diversity or microbiome studies. Next slide, please. Now, let's look at how users can explore the dataset. There are 2 main ways to navigate. First, users can explore the data by matrix, by country, or by combining both options. For example, after selecting a specific matrix, such as environmental or human, they can choose a particular compartment and access more detailed visual information about the analyzed substances in the compartment. Alternatively, users can select a specific case study site or country This allows them to dive into all analyzed substances across different matrix within that site. So depending on the research question, users can either focus on one matrix across countries or focus on one country across matrices. This flexible navigation allows different types of comparison. Next slide, please. This slide shows the summarized way of exploring the dataset. At the matrix level, users can access total pesticide concentrations, number of detected substances, and most frequently detected pesticides. At the country level, users have access to heat maps. These heat maps provide an overview of detected compounds across different compartments within each matrix. By clicking on a specific compartment in the heat maps, users can further explore the number of detected substances, concentration levels, sample level information within the country. So the tool allows users to move smoothly from a broad overview down to detailed information. To conclude my part, the case study site results tool transforms a very large and complex monitoring dataset into an accessible and interactive platform. It allows users to explore pesticide occurrence across matrices, compartments, and countries in a flexible and transparent way. Thank you very much.
Hello, good afternoon. I'm Paula, and I will present the ecological risk that we assess in the samples from the case study sites. So I will start by briefly introducing the risk concept here, and some of you might have seen this picture before. So we have a hazard, which is something that can potentially harm you. But that hazard will only be a risk if you're actually exposed to that hazard. So in this example, the hazard would be the shark swimming in the sea, and the risk would be if you are swimming in the sea with the shark. So we can think about the pesticides in the same way. So the pesticides would be the hazard because they can potentially harm the organisms in the environment. And the risk of the pesticides will depend on their exposure. So if you go to our toolbox, you can see the ecological risk of the case study sites in the left menu, and that will lead you to the introduction page. And there you can see the brief explanation of how the ecological risk was assessed. Next one, please. So for exposure, we consider the pesticide concentration in soil and water samples from the case study sites that were the results that Rosita showed. And for the hazard here, I called effects, and we use toxicity threshold that's called the NOEC, which is the no observed effect concentration. And this is a concentration where no effect on the organisms is expected. And these values were taken from the PPDB database. Next one, please. And then we calculated a risk quotient. For the terrestrial ecosystem, we used the concentration that were measured in the soil, and we divided that by this toxicity threshold, which is the NOEC for earthworms. And in the aquatic ecosystem, we used the concentration in the water samples, and we divided by the NOEC in fish. And what we did, we sum up these risk quotients of all pesticides detected in each sample. So the sample would have a cumulative risk quotient. And within the European legislation, the concentration of these pesticides in the soil should be 5 times lower than the NOEC values for earthworms. And that's why we used a safety level of 0.2, below 0.2 for the risk quotient here. In the case of the aquatic ecosystem, the concentrations in water, it should be 10 times lower than the NOEC for fish. So in this case, our risk quotient to have a safe level should be below of the value of 0.1. Next one, please. And then in the left menu, you can see the results for soil and water. So when you click there, it will open these two plots for you. In the first one, you have the sum of the cumulative risk quotient for each sample. So which dot represented there would be one sample. And then the red line represents the safe limit of 0.2. In the graph below, you have the percentage of samples by risk category. So you have samples in gray that are samples that had no residue, so no risk. In green, you have the samples that had the cumulative risk quotient below 0.2. And in purple and dark purple, you have those cases where the risk quotient exceeded the safe level. Next one, please. If you click in a country, for example, here we have Portugal, it will open a second page where more detailed information for that country is shown. So in this plot, you have each column representing one sample and all the pesticides detected in that case study site. Then you can see the risk quotient for each pesticide in each sample. In many cases, the high risk were related to the presence of one pesticide, as you can see the purple colors. But if you look at the last row, you have the cumulative risk. And for example, that first column on the left, that sample, all pesticides had their individual risk quotient below the safe level of 0.2. But when you sum them up, the level exceeded the safety level. So this is to show the importance of considering the pesticide mixtures in the risk assessment. Next one, please. The same way you have the results for the water samples. And here in this case, only one sample exceeded the safe level of 0.1, as you can see in Argentina. But you can also click there in the different countries and you will obtain this more detailed information of each sample and the pesticides that were there and the risk quotient. So next one, please. Okay, so now I will continue showing another part of our toolbox. And this is related to the toxicity results. And in this case, we conducted tests in the lab where we added the pesticide mixtures to our soil or water, and then we exposed the animals under lab conditions. So next one, please. So in the main menu on the top, you'll find this toxicity/ecotoxicity, and you can click in the ecotoxicity results. Then you will be open to the introduction page where we'll explain how the mixture selection was conducted. So briefly, we'll show we have these 11 mixtures, each one representing one case study site. And then we select the top 5 pesticides of most concern in each case. And this was selected by having the risk profile of the pesticides and their frequency in the samples. And then we select the concentrations to use in the test. So we have one control, that was the pesticide-free soil or water. And then we have the MAC, which were the measured concentration of the pesticides in the case study site samples. So these are very realistic concentrations. And then we chose to use the PEC concentration, which is the predicted environmental concentration, and those we took from the EFSA reports. And to test worst case scenarios, we use 3 times or 5 times the PEC concentration. Next one, please. Here's an overview of the species we used in those tests. You can see in brown, there are the standard species that are the species used in the risk assessment of the pesticides. And in green, we have what we call the SPRINT indicators, which are species that are non-standard. And we conducted tests with them too to have an idea how the mixtures would affect those species too and have a comparison. Next one, please. In the left menu of the toolbox, we have the methodology for soil and water. And when you click there, you will see a brief description of the test. And in case the test was a standard one, the guideline that was followed will be referenced there. And you have pictures of the experiment for illustrating how they were conducted. Next one, please. And then we have the results section also separate for soil and water. And here you have the visual representation of those results. So you can check which mixtures were more toxic to which organisms and the concentrations. And for all these results that have been published, the link to the publication will be there. And this will be updated as the publications are out. Next one, please. Yeah, so that was.
That is it. Thank you. Thank you very much, Rosita and Paula, to indeed show us the results that we got from the field campaign and also in the lab mixture testing. We do have now time for a couple of questions. I would like to invite then Rosita and Paula to turn their cameras on so they can indeed share their thoughts with you about this. So I see in the chat there is a question from Silke about can one apply this toolbox also outside the mentioned countries, for instance, Uganda, Kenya, Tanzania, Nigeria? Jacob, maybe?
Yeah, perhaps I can answer. So yes and no. So by the principle, it's definitely applicable to other regions than the Europe and Argentina, which were the primary under the primary focus. However, the functionality of the tools depends definitely on the underlying data. So if there are the compatible data, the sufficient accessibility, resolution, and so on, the most of the tools can be translated or replicated into the other regions. So we take this toolbox mainly from the global perspective as inspirations, how the things can be done, what approaches we used, how we combine the data to get to the predictions, models, maps. The second thing is that we are active in this going globally. So part of the activities of the SPRINT project is also comparing the toolbox and individual tools and their usefulness and applicability for low and middle income countries. And that's done by FAO. FAO is also a partner of the SPRINT project. And one of the last deliverables is focusing on that. So there we are in detail comparing Toolbox and FAO pesticide registration toolkit, despite they have a bit different focus, like the Toolbox as was and will be emphasized is more focused on post-registration phase, while the pesticide registration toolkit of FAO is for registration phase. There are also common things, and that will be reported later in the comparative report.
Yes, thank you, Jacob. Indeed, stay tuned. We are indeed working also about this FAO indeed report about global application. So yeah, stay tuned via our SPRINT website as well about the latest developments and publications. We have still time for one
if I can
yes?
add just some results are definitely applicable everywhere, and these are, for example, the toxicity, ecotoxicity results, the wind erosion model, and so on. So these are the tools that are not based on any kind of geographical position. So I think also many tools are useful for everyone globally.
Yes, thank you. Okay, thank you. Well, we have time for one more quick question. I see another one in the chat that is about why was Argentina taking up amongst European countries? Maybe I do that, I guess.
Please do.
So, indeed, in the SPRINT project, well, it's mostly an European project focused on European agricultural practices, but considering the important role of soybean production in Argentina coming as feed to Europe, we had included also a case study site in Argentina. So that altogether, we could have indeed this One Health approach and One Health assessment, not only on ecosystem, but also livestock and human impacts. I hope that in a nutshell answers, but again, we also have detailed analysis about how Argentinian results relate or not with European findings. So in all our publications, we are also very mindful about some differences in practices and regulatory aspects between Argentina and Europe. Yeah. Again, if there are any follow-up questions, leave it indeed in the chat. We will come back to all of it to make sure that everyone gets an answer. Looking at time and to don't steal time from other tools and other tool authors, I would like now to move into block 2 of this event that covers field and national maps and then risk maps and also model library. So I would like to give the floor right away to Arthur, and then it will follow up with Paula and Danielle. Arthur?
Thank you, Vera. Hello, everyone. So I will try to introduce briefly the concentration maps tool available in the toolbox. Next slide, please. So the first step in preparing pesticide concentration maps was to compile publicly available pesticide usage data and crop-related data. This data were then compiled for individual fields by matching crops grown in a given year with pesticide usage data. In this way, we were able to create these generalized mapped usage patterns, or GMAPs, at high resolution for several EU countries. Such GMAPs are crucial inputs for modeling concentration of pesticides in different environmental media. Next slide, please. So the quantity of pesticides in soil is calculated using specially distributed input data, such as crop interception factors, soil-related variables, and pesticide application rates. The main entry pathways for pesticides are direct application to soil, surface runoff, and spray drift to adjacent river bodies. We adopted a modeling approach in which processes driven by land characteristics directly affect how much of the pesticides applied on a given crop enter agricultural soil and adjacent streams. In more detail, the concentration in surface water is obtained by taking into account farm-specific application loads and river catchment-specific discharge. We produce many maps, and they are available in the toolbox, in searchable web applications, at several spatial scales, such as individual fields for many districts, and also for entire countries.
Thank you, Arthur. So now moving to the national maps, there we used two existing models. We compare different modeling approaches. As Arthur just mentioned, so when they're spraying, during spraying, there can happen drift, pesticides escape the field, and then might be consequent volatilization. We used there called the Gaussian tilting plume model based on the RT drift equations. It's a peer-reviewed paper published. And for volatilization, we used the Pearl model equations for soil and volatilization from canopy. And the model had a probabilistic component because here we were creating maps, but at a certain moment we actually, although we had pesticide usage across the year, we don't know when a farmer applied. So we took into account conditions where a farmer could have applied, and we actually simulated all the possibilities at the daily resolution across the year when a farmer could have potentially applied. And what you see here on the field configurations beneath the slide, we tried different configurations of source estimates. So in this case, putting the sources at the edge of the fields or at the center of the field. And this, of course, creates different outcomes. And I think that's one of the outputs also of this tool, is because it's a lesson learned on the different methodologies and approaches used. Next slide, please. So what you see here is one of the outputs. This is a tebuconazole air concentrations, nanogram per cubic meter, downwind of each individual field. And of course, behind this map, it's a big data set, but for visualization purposes, we averaged at 1.1 kilometer grid. Next slide, please. We produce similar maps for Czech Republic. In this case, you have glyphosate air concentrations, also downwind of each individual field, average at a 2.5 by 2.5 kilometer grid. And next slide, please. And finally, and just taking a bit more time on this one, because here we did try different modeling approaches. This is astamiprid air concentrations, also averaged at the 1 by 1 kilometer grid. But in this case, we actually simulated the 5 most occurring combinations of meteo conditions at multiple downwind distance and sideway distances, which basically the data set behind this is 8.5 million rows of concentration points across Denmark. And of course, this data in the end will be available to download once the publication is published and peer reviewed. Next slide, please.
Well, thank you both, Arthur, Danielle. We will have opportunity for questions shortly. So if you do have indeed any questions about this modeling exercise, leave it here in the chat. We come back to that. First, I will like to give the floor to Paula to talk about also other type of maps, in this case on ecological risk maps, please.
So in this case, we did ecological risk maps for soil and water compartments. Next slide, please. So on the top of menu of the toolbox, you can find where the ecological risk maps are, and this will open the introduction page. There we have a brief explanation of how these maps were created. So again, we use the same concept I explained earlier. So risk will be a combination of exposure and the effects. So for exposure, in this case, we used the modeled PEC concentrations in soil or surface water. And this methodology was the same as presented by Arthur for the field resolution concentration maps. We used Czech Republic as an example for these maps. Between 18 and 19 pesticides were selected for each of the compartments. And the PEC concentrations were calculated as a time-weighted average over a period of 56 days. And that counting from the first application, one-time application in July of 2021. And for effects, we used the no observed effect concentration, NOEC, as explained before, this toxicity threshold. And we gathered this data for different species of earthworm and fish. We took the data from 3 different databases, the PPDB, the EFSA Open Food Tox, and the US EPA Ecotox. And the lowest NOEC value we could find, we retained for our calculations. Next slide, please. In the introduction page, you can also find more information of, for example, how we select those pesticides. We started by having the used data of the year 2021 in the Czech Republic in tons per year. And we also used the chemical and ecotoxicological properties. So with these 3 data, we make a selection of the pesticides. There you can also see how the risk quotient were calculated. It's very similar also to what presented before, but in this case, we use the PEC values that were modeled divided by the NOEC. And we also did the cumulative risk by summing up all the pesticides that were occurring in a specific field or in some one river segment. Next slide, please. Under the introduction, you can click in the ecological risk maps, and there we'll have this map browser where you can select a location which will be different district in the Czech Republic. You have the compartment, either soil or water, and you have chemical will show the list of the pesticides that were modeled. But you can also choose cumulative would be the cumulative risk quotient for all the pesticides in the area. Next, Jane, please. If you zoom in the maps, you can click on a specific field, and that will open more detailed information for that field. So for example, we will have the type of crop that's there. You have the cumulative risk quotient value, and you also see the top contributors for that value. Next slide, please. In the same way, you can have the water compartment. You see the different river segments. Next, please. And in the same way, you can also click one of the river segments, and that will have a more information on the cumulative risk quotient and also on the top contributors for that area. Next.
Thank you, Paula, very much for this presentation on the risk maps. I encourage all the people with us here to also go to the website and explore. We are giving here some examples on how indeed this tool can be used and the type of outputs that you can find, but there are others. Of course, we did some highlights, so do use the website and do explore, and if there are a more practical or content-related question, don't feel shy and just leave it here in the chat, because shortly after this blog, we'll have also opportunity to address them. Yeah. Okay. Jakob, please.
Yes, thanks, Vera. So the next tool are the pan-European maps, and this tool brings a high-level overview of pesticide pollution across the entire Europe, and it visualizes modeled annual average concentrations of about 120 substances in 4 compartments: agriculture soil, natural soil, freshwater, and air. And I also have to say that this tool was created by Dr. Farshad Sohailifard from DTU and his colleagues. However, unfortunately, he cannot be with us today, so I will present the tool instead of him. Next slide, please. So how these values in the maps were obtained. We combined the global data on real pesticide usage from 2016 to 2020 that shows where and how the pesticides were used. These data are commercial data from a commercial agroeconomy company. And these global pesticide usage data were then spatialized using global crop production maps at a resolution of 5 to 5 arc minutes using the spatial production allocation model. Next, then we used the emission distribution model called PEST LCI consensus, which uses the information about crop growth stage, and pesticide application methods, and it derives a set of environmental primary emission distributions into air, soil, crop, and water. And it's based on a mass balance after the pesticide application. Next. Then the geospatial Pangea model, the global geospatial Pangea model, was used to build a set of grids for relevant compartments using the Pangea GIS engine and a set of environmental models. So this is called geometric system, and all these georeference data were projected in this system. Next. The last step before getting the maps was refining of terrestrial and atmospheric grids, resulting in multi-scale grids in Pangea. So what does that mean? So we aim to get a high resolution in the areas of interest. And this prioritization of the areas was based on ecotoxicity impacts combined with ratio between the population to freshwater volume. Next. So then the results are these pan-European maps showing the estimated pesticide concentrations. Next. In soils. So for example, here you see the example of acetamiprid in agriculture soils in Europe, and the polygons are the river basins. Next. Then the concentrations in freshwater of the same substance, and then next in air, in atmosphere. Here you see that these rectangular polygons, their derivation was a bit different than for the river basins that were used for the soil and water results. So with this slide, I really want to remind that the tool shows model predictions. Definitely, it's not showing the real measured values. These are the estimations that were based on real usage data, combined with real information about crops and so on, but not in any way the measured values. And we really believe that this tool helps policymakers and researchers to identify especially the critical zones, so the zones of higher risk and environmental footprints of pesticides use at the European level. And these maps can start or can help to serve as a strategic starting point for targeting monitoring campaigns or some further local investigations and so on.
Thank you. Thank you very much, Jakob, for presenting the tool. I also see that Olivia is with us, so if indeed there are any questions also about the underlying assumptions and approaches done with this tool, again, just leave it in the chat. We'll come back to that just after Danielle's presentation about the model library. Please, Danielle, the floor is yours.
Thank you, Vera. I'm just going to discuss the model library. So this is a collection of different available fate models. Some were used within SPRINT, many were not used within SPRINT. Next slide, please. So how we came to this collection of models, this was based on an initial scoping review, and also after that expert input in case we missed some models. The library might not be complete, I should stress that. And the idea is, as you can see from my input on future community input, is that you can keep on adding. There's contacts on the model library tool that you can send an email with the relevant information. And in this case, it's the model named, the keyword. In this case, we have 4 keywords: dispersion in air, if it's focused on soil, water, if it's a multimedia model, the main purposes, the main output, if it's open source, and a download or contact person. And then you can also, in this tool, go and search by keywords. You can search if it's open source or partially, and you can also see if it was used in SPRINT by clicking used in SPRINT and seeing if it was used, yes or no. Advance, please. So here are the links. In yellow is the FATE models library. And also within the FATE models and below, there's also a model called SWIPE. So SWIPE was a wind erosion model developed within work package 3 of SPRINT that is now made available as an application. And there you can play around basically with parameters such as particle size, wind direction, wind speed, and see how particle phase pesticides are carried by the wind at different distances. So also feel free to access at the wind erosion model link below to explore this tool. Advance, please.
Well, thank you. Thank you all for these presentations. I would like to invite to all the speakers of this block 2, so Arthur, Danielle, Paula, Jakob, to turn your cameras because we do have some questions. So, okay, I will just start selecting them a bit randomly. I see one of them, and from Gustav Bostrom, I believe it relates to the first maps of Danielle and Arthur, that says, what made you choose the Netherlands, Czech Republic, and Denmark for the national maps? Did you identify any other country where you could do the same approach, or is the lack of data in other countries a restrictive factor? In that case, which type of data is lacking? Arthur, Danielle?
Arthur, you can take it.
Yes, sure. I think the most decisive was crop, so data granularity. So too much crop maps with PPP usage data. We needed to have homogenized data. So in terms of what crops match PPP usage.
Yeah, and if I can add to that, just we did do inventory of PPP data. There's a publication coming soon on that, and that also served as a prioritization for here. So that was important.
Thank you. I see a kind of a coupled question to this that says, really interesting heat maps. Are you planning any cross-validation of the model values based on measured results? And I believe here also Arthur, I guess, could also add.
Yes, exactly. So that's what we are working on now. So we had a lot of model data, and we have also from the early part of the project a lot of measured data. So now the next step is to have an idea how our model performed.
Thank you. I get the impression that.
So if I can, that's also different for the different levels. So I think what Arthur just said is relevant to the national and field resolution data, where in the planned or finalized publication there is this cross-validation with the real measured values. I can say for the pan-European maps, Olivier asked to be unmuted. I don't know if this is technically possible. But,
I tried.
Yeah.
I just tried. I don't know if it's working.
It looks like
Yeah.
Sorry, I don't have the Sprint background. Just Pangea, and we have a global now pesticide inventory of application inventory. So Pangea has been now applied worldwide, and we have first results and maps worldwide. So if people are interested, publication will come in a few months from now, but at world level. And in fact, underlying, we could go and question about normalizing per kilo applied. In fact, many of the factors, we first look at them for kilo applied, but then it's interesting to look how much is applied. I mean, both are important. So for the resulting concentration.
Thank you, Olivier, and very good to have you also here with us. Thank you. Well, I think we still have time for at least one more question. I see one more in the chat here that relates about data. Well, we have talked about models and underlying data. So Beatrice says, can you discuss challenges regarding access to data on pesticide use from farmers? In Canada, for example, there is only pesticide sales data, not use data. How was data access for the maps that you have created then? Thank you.
So we use only usage data, right? We didn't base our estimations on sales.
Okay, well, it's a straightforward answer indeed. In our field campaign, we did have interviews to farmers to collect some data about their application scheme, which help us a lot to interpret the chemical findings. But indeed, for this type of modeling exercise, we had to also follow other type of approach, and indeed, sales data was it. Okay, Jakob?
Yeah, maybe I would like to emphasize what Daniel already mentioned, that there are two upcoming publications. One will be on an inventory of the usage data across all the Europe, and the other one about plant protection products registers across the Europe. So these two inventories will address how the data differs, what are the resolutions, accessibilities, and so on. So based on these, we also prioritize the countries, but the inventory will give you the impression which countries can be considered as a data rich and which other countries have a not so good data. So to create similar tools like we did will be limited.
Thank you. Okay, Olivia, please, you have your hand raised.
Yeah, just to say complementary, we've been working with Yuetang on a global inventory based on sales data, but then extrapolating for countries, we don't have so much data, having machine learning to try to get a first estimate. But these are first estimate, we have to acknowledge. It doesn't replace first-hand data.
Thank you. Well, I think I can speak on behalf of the consortium here that we did have quite different approaches and quite some challenges around data, but it also highlights a lot of potential. So some of our lessons learned coming afterwards are also related indeed to data and access to data. Okay, maybe one last question I see here for Martin. Says, I think it will be interesting to normalize environmental concentration to application rates. Otherwise, the data show the compounds that are applied at higher doses also lead to higher environmental concentrations. What is your view on that? Anyone would like to discuss about this normalization
So
application?
You want to take it, Jakub, or shall I?
Yeah, I would just simply said that both data are very important, but from the perspective of possible risk or effects on organisms and so on, we are interested in these concentrations regardless if they are results of some time accumulation or fresh application, whatever. So this is what is there, and we are interested in these.
Beautiful. Do you have anything to add, Danielle? No? Okay, well, thank you all for these questions. And again, if these answers also trigger follow-up questions, don't feel shy, just leave it us in the chat. We have the opportunity to come back to it later today or even via email. I would like then to move to block 3 of these presentations, which will cover dietary exposure, health impact, and toxicity results. So I will start by giving the floor to Shiva and Danielle, and then we go from that together, if that's okay. Shiva?
Sure. Good afternoon, everybody, and thanks for joining to this session. I'm very pleased to be here to introduce you the human dietary exposure tool in a SPRINT Toolbox. Next, please. Humans may get exposed to pesticides by 3 different pathways, which are respiratory, oral, dermal pathways. Studies show that for the general population, the main route of exposure to pesticides is through food consumption. Next slide, please. In this tool, we presented the estimated average amount of pesticides European adults are likely to get exposed to through their diet. Next slide, please. To be able to calculate dietary exposure, we needed 2 databases. First, database of pesticide residual concentrations in food commodities. For this, we used the raw data from annual food control activities on pesticide residues, which carries out in EU member states, Iceland and Norway every year. These data are available since 2011 and have 33 European countries involved. We use the data for the years 2011 to 2020. And the second data we needed was a database of food consumption. And for this data, we used the raw primary commodity database. Both of these databases are open source and available for public to download from related platforms. Next slide, please. For our estimation, we selected 10 most commonly consumed primary food commodities based on RPC data. To deal with the residue concentrations lower than LOQ, and imputation was carried out, and to achieve an accurate output for only active substances fulfilling two conditions were selected. First, having at least 15 observations above the LOQ, and having data not more than 90% of the whole data. Finally, imputation was performed for the selected pesticides using maximum likelihood estimation. Next slide, please. And average dietary exposure was calculated then by multiplying the average of residue concentrations, of course, after imputation, by the average of adults consumption for each European country. Next slide, please. The output of these calculations are presented in SPRINT Toolbox in the Human Dietary Exposure section. By clicking on the pesticide residues in food or dietary exposure tab in the left side menu, users can see the maps of average pesticide residue concentrations or average adult dietary exposure maps respectively. Next slide, please. In the tool, there is possibility of filtering the maps for food commodity, pesticide, and the year to see the corresponding maps. Here, you can see the residue concentration and dietary exposure maps for chloropyrifos in apple for the year 2011 as an example. And in the left side, you see the concentration map, and the right side dietary exposure map is illustrated. The maps gives the opportunity to the users to follow spatio-temporal trends for each food commodity and pesticides. Next slide, please. The numeric values are also available in the toolbox right below the maps for more exploration, and also there is possibility to download the data in the CSV format. And also ADI values are presented in the toolbox for risk assessment purposes. Thank you.
Thank you.
Danielle?
So we also looked into farm animal exposure via feed intake. Next slide, please. Advance, please. And here we had basically three data sets. So we had the pesticide in feed data that was analyzed in the case study sites, where you can find this information within the toolbox, as mentioned by Rosita. Then we did a scoping review to get variability on body weight distribution per farm animal type. Initially, we had used the European Food Safety Authority average values for our assessment, and then we decided to actually try and see what would happen if we actually vary the body weight in our assessment. And we did the same with the feed intake. Instead of just taking an average feed intake, we did a scoping review to look at what would be the distribution of total feed intake per farm animal type. We did this for 4 different types of animals: sheep, goat, cow, and chicken. And in this case, what you're seeing in this illustration is a density plot outcome dietary exposure, in this case feed intake exposure in the x-axis for cow and azoxystrobin. We then compared all calculated exposures with health-based guidance values, most of them derived by the no adverse effect levels for mammals, and birds for the case of chicken. And you can find in the toolbox this approach, as well as comparison with the health-based guidelines value which in this case was below for all except for chickens for permethrin, and in 3% of the population was above the health-based guidance value for chronic exposure, which might not be as relevant given the lifespan of chicken. So you can explore this in the toolbox. It's based only on the CSS data, therefore, please interpret the results with care. Thank you. Next slide, please.
Yes, thank you both, Shiva and Danielle. I would like to ask Charlotte to put once again the link to the toolbox in the chat. I see that there are still people connecting. We are now 99 people online at the moment. Thank you for being with us. And I also saw a question about if the event is being recorded. Yes, it's being recorded, and it will be shortly after today made available in our Sprint YouTube channel. So you can go back to the tools that you did not have the opportunity to join or to revisit and so on. Do use the link that Charlotte, thank you, just put in the chat, and explore indeed these functionalities that are being displayed and others, because there are more than the ones that we are showing here today, okay? And leave questions if you like to have clarification on the tool or content underlying this tool. Without further ado, I'd like to give the floor then to Arash about the health impact assessment, please. I may have lost the speaker. No? Arash, can you hear me? Yes, Anki is here. I see at least.
I see it just before, but otherwise I can, of course, take over.
Or we can, if that's okay, we can also start first with Simona, and then we see if Arash joins. Is that, or not? Sorry, I don't want to, because there is an order in the slides. Don't want to mess up.
I think it's also fine to continue. Okay, I will present the part on the health impact assessment. Yeah, thank you. Can I have the next slide, please? So in SPRINT, we did not do any epidemiological study, but we did have a look at human health impact. And what human health impact assessment does is essentially it tries to combine 3 different types of data. It looks at population exposure distribution, that looks at existing exposure response relationships from studies, and then it calculates the number of debatable cases in a population. Next slide, please. So the most important part here is, or the most complicated part is to extract the exposure response functions, because these need to come from external data. What we did here was to really do a complete systematic review, searching and then reviewing these studies to try to assess established functions from the literature. For the exposure assessment, we used data that came out of SPRINT. Next slide, please. We looked at 3 different pesticides, and in the end found only existing data for glyphosate. We used for glyphosate 2 different health outcomes that we assessed. The one is that we looked at the incidence of metabolic syndrome, or the prevalence of metabolic syndrome. We used for this study the urinary levels that have been evaluated in the SPRINT case study sites. You heard the results before. Can I have the next slide, please? We found 2 different studies that had a look or assessed the effect of urinary levels of glyphosate on metabolic syndrome. And we meta-analyzed these data, and the overview is shown here. We also provide as a tool in the toolbox the R code on how to actually do these kind of calculation steps. Next slide, please. So the interesting part here, we did a calculation for the whole country of the Netherlands. We used the urinary levels, we used then these combined exposure response functions, and we used existing health data from our health registries. And what you can see is that for a pesticide that has a very high prevalence of exposure in the population and an outcome that has a very high occurrence in the population, you can get to very, very high numbers of people who are affected. So this is a relevant insight that came out of this calculation. However, this exposure response function, because it relies on only 2 studies, has of course very, very high uncertainty. Next slide, please. Another example that we calculated was for a birth outcome in newborn. It's a defect of the heart, an atrial septal defect. Next slide, please. So in this case, the underlying study that we applied for the exposure response function had a different way of the exposure assessment. It used the amount that was used of a pesticide in the environment of people, of the pregnant women, and then assessed the risk on the newborn children. We used essentially the approaches that were shown before, based on the modeling in SPRINT, to calculate what that would mean in terms of attributable cases. In this case, next slide, please. In this case, what you can see is that this number of cases, attributable cases, for one country, 1 year, is actually relatively low, and that is because the outcome is relatively rare. So even though we looked at the same type of exposure in the same country, again 1 year, the number of cases is much lower. So this is the effect essentially of this outcome being quite rare. Next slide, please. So the bottom line is that these numbers are incredibly highly uncertain. And this is not because of only of the exposure, but primarily because there are so few studies that actually evaluate these exposure response functions. Nevertheless, what we can see is that high exposure plus high outcome can relate to very, very high public health impact. And this means that in essence, these kind of associations need to be evaluated much more frequently. We have in the toolbox provided the tools, the steps really in detail on how such a health impact assessment needs to be performed, including the systematic research of the literature, the review, the combination of these exposure response functions, and how this can be applied to calculate impact of health in a population. Next slide. We also provide 2 different Shiny tools. So these Shiny tools are there to play around with simulated numbers. So here you can put in numbers to understand also effects of multiple pesticides on health effects. Next slide, please. And then to calculate the according attributable cases in the population. Thank you.
Thank you very much, Anke. Yes, beautiful. So we go now for the last presentation of this 3rd block, actually the last presentation of the day. So I would like to invite everyone to leave any questions that these presentations or previous ones that you like to raise in the chat. So we come back to that shortly. And without further ado, and a bit ahead of time, I hope that I have Simone with me.
Yes.
Yes, okay, please, the floor is yours on the toxicity results.
Thank you, Vera. So good afternoon, everybody. I'm Simone, and I will be presenting the latest toolbox for today. So the tool toxicity testing provides an overview of the main outcomes from the toxicological tests conducted within the SPRINT project. And all the experiments described in the toolbox have been published as research articles or are in the process of being published. So in the next slide, please, there is a scheme of what was performed for this assessment. And in order to provide valid information on human health and to refine, reduce, and replace the use of laboratory animals, we followed a step-by-step approach, selecting the main pesticide of concern to be tested on rodents only at the end of the project. And this multi-step approach divided in tier 1, this one, tier 2, the second one, next slide, and 3, tier 3. This multi-step approach is based on key ideas from 2019 EFSA guidelines. So the next slide, please, provide an overview of the toolbox. Users can follow all the 3 stages of the third approach by clicking on methodology to get an overview of the main procedure used for this assessment. And to learn more about each step, users can click on results tier 1 or 2 or 3 and consult the main result obtained for each phase of the research. So moving to the next slide, tier 1, we used the available data on around 200 pesticides collected from SPRINT case study site in order to perform in silico modeling aimed at identify 20 pesticides of concern ranked by occurrence. Then the selection of the top 10 pesticides to be studied in vitro in tier 2 was based on other assessment. So in the next slide, we have the tier 2, the second step. So in tier 2, this list of the 10 main pesticides was tested in vitro individually, and also as a mixture of the top 3 or top 8 pesticides. Both EFSA standard tests were used together with the new SPRINT indicators in vitro. The EFSA standard test included a cytotoxicity test and also a micronuclei test to assess whether the pesticide could kill cells or damage DNA. And the new SPRINT indicators used in tier 2 in vitro were based on a kidney cell test to detect kidney damage and also on organoid test to study more complex responses. In general, the pesticide tested and their mixture significantly interfere with the cell viability, vitality, and also DNA. In the next slide, there is the final step of the tiered approach. In tier 3, the final stage of this stepwise approach, the top 3 pesticides and a mixture of them were tested in different in vivo assays. The 3 compounds selected were representative of the 3 main categories of pesticides: the glyphosate herbicide, etabuconazole fungicide, and acetamiprid and insecticide. All pesticides were tested at dosage range considered safe, that is between the acceptable daily intake and the NOAEL, so the level with no adverse effects in rodents. For tier 3, also in this case, we used both the standard EFSA test and the new SPRINT indicators. For the standard EFSA test, a 13-week toxicity study was conducted on male and female rodents, starting exposure from prenatal life in order to mimic human exposure. The new SPRINT indicators assess various aspects, such as microbiome evaluation, but also behavioral studies on mice and rats, together with electron microscopy of kidneys. And as you can probably imagine, it's not possible to summarize of these results in 5 minutes, but I strongly encourage you to consult the toolbox to get a clearer idea of what was performed. But in general, we can conclude that this kind of stepwise approach was successful and allowed us to optimize resources on pesticide of greater concern, since all the pesticides that have been prioritized in tier 1 and tested in tier 2 and 3 showed at least one positive signal. In general, pesticide mixtures proved to be more detrimental, and also in general, the new models used in SPRINT, so this new SPRINT indicator, have proven to be sensitive and relevant for several toxicological endpoints of interest. Thank you. Thank you
Very much, Simona. We're perfectly on time, so I will go right away to Jakob to do some wrapping up from all these presentations. And after that, we will have some time for questions. Yeah, please, Jakob.
Yeah, exactly. So allow me to do some concluding remarks. So in the past 70 minutes, you learned what the different tools in the SPRINT Toolbox provide, what they can do, how they function, how they look, that you can also explore yourself on the link, which was several times placed in the chat. Now I would just like to summarize. Next slide, please. So that's the maybe more detailed scheme of all the tools that you have heard about today. Next slide, please. I would like to summarize the benefits of the toolbox for various stakeholders. So we really believe that the SPRINT Toolbox provides supporting tools for improved access to the data on pesticide occurrence, environmental fate, exposure, and toxicological or toxicological effects. Also showcasing how to process and visualize the big pesticide-related data on real examples of this data from selected European countries, and from also this national level to the European scale. Also, it provides the improved and geographically explicit risk assessment of pesticides, and not just individual substances, but the mixtures. As I said already, we think that major use is in the post-registration phase, in the regulatory context related to the sustainable use of pesticides. We also think that these tools can support information-based agri-environmental governance and decisions. Concretely, they can help, especially the maps, can help to identify the priority areas where there is some peak or accumulation of pesticide exposures or cumulative risk. And this can help to prioritize management interventions or target the monitoring campaigns, by the way, which will be obligatory soon in Europe to monitor the soils, including pesticides. So this can help to design the monitoring plans. Then it's indeed the support for designing and prioritizing pesticide reducing strategies, let's say the removal of the substances or substitution of the substances, because our tools can indicate which substances appear to be repeatedly problematic. Then for monitoring, reporting, and evaluating mitigation strategies, simulating some withdrawal of the substances, how, for example, the exposures and cumulative risk will change if a given substance is not used anymore, and overall supporting the planning of farm-to-fork related policies for the sustainable use of pesticides. Next one. So we have also the data-related take-home message. So what we have demonstrated by multiple tools, that the big data related to pesticides, like usage statistics or authorization data or resolute crop maps or hazard databases, have really enormous potential to feed the tools for decision making and help for pesticide reducing strategies. We also showcase such tools in the SPRINT project, so that's the content of the SPRINT Toolbox that you have learned today. And we also have some recommendation or wish that international standardization and increase of resolution and guarantee of public availability of such data are really needed to be able to do such, to prepare such tools. Thank you very much.
Thank you, Jakob. I invite everyone also that if you have any question on this vision that we have, or if you have additional applicabilities of our toolbox, to leave us also a remark about that in the chat. I would like, we have now like 10 minutes, a bit more than 10 minutes for questions. I would like to invite all the speakers of this block 3, but actually from previous blocks as well, to turn off their cameras so we can go through these questions all together. And if something pops up, just let me know. I will pick up, well, maybe starting, I see here, for instance, a very targeted question to Danielle about from Martin saying, regarding the model library on the SPRINT website, can you provide further information on SWIPE model to predict wind erosion on particle phase pesticides? Will the code be made publicly available?
Yes, thank you for the question, Martin. Yes, eventually it will be publicly available. Actually, the description of the model as well verification is on deliverable 3.2, publicly available on the SPRINT website. We are working on the publication now to be peer reviewed, and then the code will be made available. The idea of the SWIPE model is that will be hosted in GitHub, and it will be a community supported model. So you can also give input, give comments on the model itself. It's quite a simple model. I'll not go into detail now, but I think it can always be improved because it's one of the first ones focusing on the particle phase pesticide and erosion. Thank you.
Thank you, Danielle. I have another, it seems like a bit more clarifying question towards Simona. There was someone in the chat, Laura, asking, what is a standard EFSA test and what is a SPRINT innovator test? Could you just clarify our terminology, please?
Yes, that's interesting question. So what we call standard EFSA tests are a group of basically OECD guidelines usually used by EFSA in the regulatory process. And for example, for the in vitro test, the OECD guidelines is the 487, and for the in vivo test, the OECD guidelines is the 408. Regarding the new SPRINT indicators are different for each in vitro or in vivo test, but I think you can have the access to the deliverable. I'm not sure, Vera, correct me. The deliverable is the 4.5 and 4.6, where there are all the indications about the new SPRINT indicators and EFSA tests for each tier.
Thank you, Simona, for the clarification. Indeed, I also invite everyone to go to our SPRINT website, where you can have access to a lot of our deliverables and publications, where a lot of details had been provided and further interpretation as well. And again, if any question comes out of it, also feel free to drop us an email at sprint@ur.nl. We are very happy to follow up. Okay, I keep looking at the questions that we have received. I see one more seems to be targeted Shiva that mentions about the dietary model. Are those models for human consumption be calibrated against information provided in EU MRL annual report? Would you please elaborate on this MRL integration in the tool?
Maybe Daniel can go for this.
Oh, for that.
Yeah, sure. So the question for me was a bit vague, but because consumption is not related to the MRL in this, so we have two separate databases: consumption database and the database of the pesticides in the different food commodities. I think just to make clear the difference here, because there's different approaches, of course. European Food Safety Authority is an approach. There's the MCRA. The difference here is that we use the different imputation method. We can fit lower boundaries based on theoretical minimum pesticide in concentrations, because we're after realistic exposure estimates and not at the risk assessment part. So really we're after the part. The extra thing, and Shiva is working on a publication to be submitted soon, is that we do a verification step where we do the predicted dietary exposure versus measured concentrations in food baskets, also done within SPRINT, which is one of the things that I think lacks a lot in the dietary exposure models, is lack of verification, because there's no comparison actually with real case of what people are actually consuming. Thank you.
Thank you. Thank you, Danielle, Shiva. Okay, I'm going through quickly the questions. Again, we want to have the opportunity to address all your questions, but if you feel that you would like to have a follow-up or more details, just let us know. Okay, I see here, for instance, about the last slide of Yakov, there is a comment in the chat saying, well, congratulations, first of all, and on the last take-home message concern international standardization and public availability of data. These are important issues of global concern and the global framework on chemicals, which has a pertinent target on highly hazardous pesticides. Is SPRINT team engaging with the global framework of chemicals?
Yeah, that's a tricky question. I don't know, in fact, know the global framework of chemicals, so I cannot answer. Sorry for that.
Well, I think the answer is indeed not yet. Indeed, we do look into the characterization of the type of chemicals, and we do also make assessments based on a hazard. And a lot of our publications look into the hazard characteristics of pesticide. But no, indeed, this is a good point. Thank you for highlighting this. We will indeed explore it a bit further.
Yeah.
Thank you. Thank you for this remark. Okay. I also see, well, still on Jacob, there was also one question here. You mentioned that soil monitoring will be soon mandatory due to the adoption of the soil monitoring law. And someone in the chat is asking, when will this monitoring become available? Is this a matter of months or years?
Yeah, so it's actually, there is some implementation period. If I'm not mistaken, 3 years from the law became valid. So it will be somewhere in 2028 where the countries have to come with some propositions, deadlines, plans, and start the monitoring. And the first reporting should be, I think, some years after that.
Yeah,
So
after
I would
that.
say not months, but years.
Yeah. Yeah, and indeed, we have also been in close contact with the GRC, which is also developing some guidance on how to make this transposition for member states on the implementation of the soil monitoring law. And in terms of our findings, so what we did find in soil, but also our prioritization indicator. So we are hoping to indeed contribute to this guidance to be provided to the member states. Thank you for all the questions. I see here also, well, I think we'll make a record that we'll manage to address all the questions. So apologize a bit for the speed, but it seems that it's possible. I see only one more, no, two. One of them is that we do have, how is this data and models that we do provide will help in developing strategies for sustainability. Do you have any thoughts, again, on more practical applications of these data and tools that you'd like to share?
Yeah, so yeah, the toolbox is definitely not just a collection of models and databases. So it has a power to support for decisions, to make the strategies for reducing pesticides or for their sustainable use. One example how the toolbox can support that is that it provides more precise data to make more precise decisions. So, for example, the maps, they can identify the hotspots, the problematic areas, but because these maps are underlined with the data about crops and usage data and so on, it can be tracked back which substances, because of which crops, brought these increased concentrations, or the risks, ecological risks for aquatic or soil organisms. So we can track back and identify and prioritize the substances based on their real ecological risks, not just based on their labeling in a current hazard index, which is used in European Union. So I would say geographical precision, then active substance resolution, individual crops resolution, and all of that can help to decide which substances will be removed from the list or substituted It can also identify the substances that are used and occurring, and at the same time not posing so high ecological risk, not contributing in the mixtures so much. So these are the examples how this can be used for a very precise designing of strategies for sustainability.
Thank you, Jakob. And I think it is also here we invited several stakeholders to use it and explore it, and indeed to see how they can also meet their needs.
Yeah.
So please explore it. This
yeah,
is
as we also highlighted, we are showing the way how to do that. So the tools are not the final fixed data shown, but they rather show how to do that if you have the data and so on. So it can be replicated if similar data at similar quality exist.
And I think you answered the very last question that was about, can it be used to other countries? Well, that it is. We do have the approach and the codes, and if you do have indeed the data, is in principle possible that you can apply it to other locations as well. Well, I see it's time now. I wanted to first of all say thank you very much to all the presenters and also my colleagues in the backhand that supported this webinar. Thank you very much for this, for the last 5 years actually, for all the work that you put into it, and also for everyone joining us and leaving your comments and your questions. It was a pleasure. We'll make the record available if you want to recatch any part, and please do explore our toolbox. And we would like to hear from you how you found it also useful for your activities. Well, thank you very much. Hope to see you soon. Stay tuned for the latest in the social media. We have here the different channels and also our website about our news. Thank you very much. Bye-bye. Mm-hmm.